All Neet aspirants, you are listening to podcast Botany for Neet by S. Ramakrishna. Keep the NCRT textbook open with you. In this session, the C4 pathway. The C4 pathway is also called hatch and slack pathway. It is a cyclic pathway like it is C3 pathway. In case of C4 pathway, it is operating in the plants which are tropical grasses, which can tolerate temperatures ranging between 30 to 40 degrees, respond to high light intensities. So the tropical grasses certainly belongs to monocot, but all monocots are not tropical grasses. If you take coconut, banana, which are also monocot but not grasses. In the C4, that is the tropical grasses, maize, sugarcane, sorghum, which are tropical grasses in which C4 mechanism operating in these plants. Next, can you differentiate the C3 and C4 externally? It is not possible to differentiation of C3 and C4 externally. How it is evident? By taking section of the leaf, vertical section of the leaf. Vertical section as per NCRT it refers to equivalent to transverse section because the leaf surface is flat and it is the section perpendicular to the surface of what it is the leaf. So it is a vertical section is equal to transverse section. Next. And the C4 plant differ from the C3 plant by having bundle sheath cell different from C3 bundle sheath cell. Bundle sheath cell surrounding the vascular bundle present in C3 as well as C4. How it is different? The bundle sheath cells which are considered as the anatomy, the Cranch anatomy. Cranch refers to breadth, ring-like arrangement of the cell. Around vascular bundle, several layers of bundle sheath cell which are large, having more number of chloroplast, thick-walled, impervious to gases. In mesophyll cells, chloroplast having grana. In this, the stroma having only it is the PEP case, but not it is rubisco. In case of bundle sheath cell chloroplast, it lack grana, agranal chloroplast, but thylakoid membranes are present which are not stacked. So it is agranal chloroplast. Stroma rich having it is rubisco. So it is having PEP case, it is having rubisco. So with the bundle sheath cells only we can recognize it is a C4 plant. We can differentiate from the C3 plant. Next, as the plants adapted to tropical climate, that is as it is a tropical grasses, it must be efficient in performing photosynthesis and also water usage efficiency should be high in these plants because the climate it is dry climatic conditions. Next, in this carboxylation occur in mesophyll as well as bundle sheath cell. So CO2 fixation separated by distance. But in case of CAM plant separated by time, night time and daytime. Here it is mesophyll bundle sheath cell. Next, in this primary carboxylating enzyme is peptidase in case of mesophyll. So primary CO2 acceptor in mesophyll cells is PEP, phosphoenol pyruvic acid, a 3-carbon organic acid. It accepts a molecule of CO2 catalyzed by the enzyme peptidase, phosphoenol pyruvic carboxylase. This phosphoenol pyruvic carboxylase is highly efficient enzyme removing the carbon dioxide from atmosphere so efficiently than rubisco. Why? Rubisco having affinity for carbon dioxide as well as oxygen. So stomata have to kept open for long time. Here peptidase having affinity with only carbon dioxide, not affinity with oxygen. So stomata need not have to kept open for long time. So there is water loss is there, but there is no severe water loss in case of the C4 plant. So water usage efficiency is high. Next, the primary carboxylation product formed is oxaloacetic acid. So PEP plus CO2 oxaloacetic acid. It is carboxylation. There is a removal of inorganic phosphate. catalyzed by the enzyme peptidase. Oxaloacetic acid formed, it is a 4-carbon acid. Next, the 4-carbon acid reduced to malate or malic acid. So redox equivalents are supplied by NADPH that generated in case of grana of the chloroplast in mesophyll cell. The enzyme it is malate dehydrogenase. So oxaloacetic acid reduced to malate, malic acid. In some cases, oxaloacetic acid undergoes transamination to form aspartate, that is amino acid. So there are aspartate formers and malate former. Our discussion is with the malate former. So malate or malic acid formed in mesophyll cell transported to bundle sheath cell. The malate in bundle sheath cell undergoes oxidation as well as decarboxylation because redox equivalents are removed from the malic acid, malate, and added to NADP. NADP again reduced to NADPH. Due to decarboxylation, pyruvic acid is formed and CO2 released is accepted by RUBP. So, CO2 acceptor in bundle sheath cell is RUBP. The enzyme catalyzing the reaction, Rubisco. The product of carboxylation bundle sheath cell, phosphoglyceric acid. So, Kelvin cycle operating in case of bundle sheath cell for formation of hexose. So, whether C3 or C4 plant, the C3 pathway is only the biosynthetic pathway to make the food. Next, whatever the pyruvic acid formed due to oxidative decarboxylation of malate, the 4-carbon acid, transported to mesophyll cells. So, pyruvate in mesophyll cells by utilizing ATP phosphorylated to form phosphoenol pyruvic acid. The enzyme it is pyruvate kinase. We have to link this type of the concept with glycolysis. In glycolysis, phosphoenol pyruvic acid is formed by which reaction? Dehydration, 2-phosphoglyceric acid to phosphoenol pyruvic acid. In this phosphoenol pyruvic acid is formed by which reaction in C4 plant? Phosphorylation. Pyruvic acid undergoes phosphorylation to form phosphoenol pyruvic acid. Next, the PEP undergoes which reaction in case of the glycolysis? Dephosphorylation and form pyruvic acid. Here, PEP undergoes which reaction? Carboxylation and forms what it is? Oxaloacetic acid. So, those are the linked questions. Next, the C4 plant are said to be highly efficient. Why? There is a high productivity. Why high productivity? It can tolerate temperature 30 to 40 degrees centigrade, respond to high light intensities. It can defeat the photorespiration process. As it defeats the photorespiration process, there is no drain on photosynthetically fixed carbon. There is a gain of the carbon only. There is no drain of the carbon. That is the carbon. Next, that's why the C4 plant are said to be biologically advanced than C3 plants. Next, so if you see the overall questions, C3, C4 and C3 plant. In case of mesophyll and bundle sheath, primary CO2 acceptor in mesophyll cells, PEP, bundle sheath cell, RUBP. Product it is OAA, here product it is PGA. Enzyme it is peptidase, enzyme it is Rubisco. What type of reactions occur in mesophyll cells? Carboxylation, reduction, phosphorylation. In bundle sheath cell, oxidative decarboxylation followed by Kelvin cycle events. Next, for formation of 1 hexose in case of the bundle sheath cell, 18 ATP, 12 NADPH2. For regeneration of PEP from pyruvic acid utilizing 2 ATP. So for every phosphoenol pyruvic acid 2 extra ATP. As in order to build 6 carbon dioxide at the site of enzyme Rubisco in bundle sheath cell, there must be 6 phosphoenol pyruvic acid. For 6 phosphoenol pyruvic acid, 6 into 2, 12 extra ATP required. So overall for formation of 1 hexose in C4 plant, 30 ATP, 12 NADPH2. How? 18 from in case of bundle sheath, 12 in case of mesophyll, total it is 30. What is the explanatory power in case of C4, 8, that is 30 ATP, 12 NADPH? What is the explanatory unit in case of C4 plant? 5 ATP, 2 NADPH. Next. So, I am signing off from this episode. We completed the C4 pathway. In next episode, you will see the photorespiration and factors affecting rate of photosynthesis. Listen with concentration, connect to the concept, because at the time of examination, recall it is going to be easy. If this approach, if you feel it is right, don't forget to subscribe and don't forget to share to the other person, those who are seriously preparing for NEET.